All right, hi everybody. So, I know a couple of you had questions about the first lab. So now, well, first lab, which has been graded already. I think everybody did it just fine on the image. I'm trying to remember. Oh yeah. So the only thing that really came up with the graph drawing, and I think that's really down to uh, how the lab handout was worded, is that when you draw standard error of the mean error bars, it's +1 and -1, right? And so some of you did +2 and -2, so your error bars looked much bigger than they should have, twice as large, obviously. But whenever you draw error bars, typically it's plus and minus one, either one standard deviation or one standard error of the mean. Pardon me, just eating my lunch. So uh, if you did that, you didn't get marked down for it because I know that you were following the instructions. The instructions were a little bit unclear. But just remember that for the future, when you're drawing graphs, it's always plus and -1, SEM, SD, whatever. Uh, Also, the error bars, I think the only person I docked a couple of points off of was because the error bars have to have those bars, right? You know, they're upper and lower bounds of the SEM in this case, right? And those are joined together with a vertical bar, right? So I think all that it was that that person kind of drew a dotted dash line, and those could be points, data points, rather than the actual error bars. So it's a little bit confusing. So when you draw error bars, even if it's by hand, they have to have the upper and lower bounds. That gives us the upper and lower limit, essentially, of that confidence. And that's joined by another vertical bar, right, with the mean typically exactly in the middle of the two, right? So just remember that for future whenever you're um, drawing drawing graphs and doing errors of the mean. Okay, so that was really the only thing that popped up for me. But I know that some of you had questions about how it was done. That's in particular why you didn't get it right. So if you want to do that all as a group now, I'm happy to do that. If you want to do it individually, um, just let me know. We can set up WebEx meetings to go over that. So if anybody got any questions about the first lab that they want to kind of go over? No? And again, I know that sometimes people don't like talking about questions in public forums, like this. So if you do have questions, let me know and I don't really want to do them by email just because it's kind of a one-sided conversation when you do stuff by email. And so I don't think that it would be as easy to kind of figure out what didn't go right. It's much better to do that, face to face, even if it's online. Okay, so if not that for that, then any questions about the second lab, the pipetting lab? Hopefully that was fairly straightforward. Pardon me. All right, and again, same deal applies. Remember, hang on. Oh no, that was actually due on Tuesday as well. Oh well, sorry, a little bit too late to be going over that. That's the pain of not being here on Tuesday to do this. Okay, so essentially what we're doing today is something it's pretty straightforward. It's a kind of lab which you've probably come across before. The intent is to really hammer home those principles of Mendelian genetics and how to assess uh, how correct those assumptions are in terms of doing your own experiments. And that's really all down to chi-squared or chi-square. Never really sure which one it is, to be honest. Test. Okay, so this is basically the lab that you would have been doing in face-to-face, but I've rejiggered it so that you can do it online. It's pretty straightforward. There's not really an awful lot to talk about. I think probably the most useful parts would be A, going through the chi-square again, just in case it didn't click first time around in class, and B, making sure you understand what we're doing in the assessment online. Right, so this one I've actually, I didn't have to make this from scratch, but it was more work than the previous ones. I've actually done it so that the questions are in Blackboard. And so essentially what we have here are two crosses, right? We have a genetic system, which is really cool, actually, in corn. And so the funky thing about corn is that these are all individuals, right? They are individual zygotes, essentially, if you want to think of it that way, of fertilized individuals, which will then potentially become corn plants of their own. And so even though it's all part of a structure which belongs to one plant, right, the actual offspring, the F2 offspring in this case, are these individual corns. So you got a huge amount of individuals on one corn cob, and you literally have a corn cob in lab. So you can do some funky genetics because all of the phenotypes are there, all the individuals are there on that corn cob. And so the two, and corn genetics is pretty fascinating too. It's one of the first organisms to have its genome sequenced because it is such an important plant agriculturally. So essentially what we have are 4 different phenotypes. We have 2 traits. They're controlled by 2 genes, right? So automatically you're thinking, oh, dihybrid cross, right? And the traits are color. So we have purple, which is here, or even, slightly purple. It's still purple. Or yellow, which you can see up here. Right, hang on, did I just making sure that I'm... Oh yeah, I did write those out correctly. And so if there's even a slight hint of purple in that kernel, it's purple, right? So you can see over here, this is kind of like an intermediate. It's not, it's purple. Okay, there's no kind of intermediate phenotype here between the two. There's no incomplete dominance going on. It's just the penetrance of the phenotype varies. So it's either purple or yellow, or, and we've got a good example here, these 2 yellow kernels, smooth or wrinkled. Right, and that's based on sugar production, I believe, in the kernel. The color is based on whether or not pigment is produced, which kind of overlays that yellow color. That's what makes it purple. So we have 2 traits, smooth versus wrinkled, which you see here, and purple versus, smoothness, and then purple versus yellow, so kernel color. And so because we have 2 traits, we have 4 different combinations, which you can see down here. And so really what I'm going to be doing is getting you to go through a monohybrid cross and a dihybrid cross. And so you get some actual practice with some of the stuff that we were talking about in lab. And so you got to remember too, and these are where the questions come in, what does this information tell you? Right, there's some easy questions in here. There's like some slightly less easy questions. So you know that the individuals that make up the parents, the P0 generation or P1 generation, are homozygous. Homozygous dominant, homozygous recessive, right? And so you need to study that. You need to answer some questions. I'm not going to tell you the answers, obviously, right? And these questions are copied pretty much exactly in the Blackboard assignment. So you go onto Blackboard. Underneath the lab handout today should be your assignment. I double checked, and God, this mouse pad goes the opposite direction. It drives me nuts. Yeah, and it's available and everything. I don't know why the exam didn't pop up. Sometimes there's so many boxes to tick that I forget. Anyway, and so if you go into the assignment, essentially you'll see these questions, and they're laid out in the same way as in here. So we have 4 exercises. We have exercise A and B, which are really down to, I just noticed there's the answer to the question in the actual picture. There you go. Study the picture. It will tell you the answer. There's some questions on just that cross, right? What does it look like? And then you're going to use this kernel, and I want you to essentially count about 50. You're going to have to, blow it up to count about 50 kernels of this, right? And you're going to have to, go this. You might need to use a stylus or something like that. Maybe print it out if it helps, and record the phenotypes of those kernels. And essentially what you're going to do is go through a chi-square test, right? So and that's going to be exercise B. And this repeated again, exercise C and D for the next cross. So the chi-square okay. So that's Andrew. I think that was you, Andrew. I don't have my chat up. Yeah, there you go. So there's 2 bits of information here. It's like a learning moment, right? Come on, there you go. That will tell you whether or not you need to worry about that. Okay, whether or not they're wrinkled or smooth. What are those 2 bits of information? There may be more. So look at the top, the title of actually telling the answer to one of the questions. What kind of cross is this? No, don't apologize. Not even a little bit. It's a really good question. But this is kind of to take you, it's a monohybrid cross. Okay, so if we're doing a monohybrid cross, how many traits are we concerned with? 1. For each trait, you have 2 phenotypes. And so what are the phenotypes that you see in the parents rather? This is the other bit of information. It's all right. You can answer by audio. They're going to be purple or kind of blackish in this case, and yellow. And so what that tells you is that this is a monohybrid cross because you only have 2 phenotypes. If you, yeah, don't worry about it. I'll speak slowly. Then you can lip read. Maybe leave and come back in. I'll see if there's. Hang on, how does this all work? All right, just give Angela a second to sign back in and see if that fixes the problem. Did that fix it? Hey, awesome. No worries. Okay, so we have 1 trait, color, two phenotypes, dark and yellow, purple and yellow. And so that tells you that I'm pretty sure that this doesn't have wrinkled corn, even though it's a little difficult to see on there. It was basically I stole this image off of another lab handout from another university. But the information given to you as part of that cross, it's monohybrid, and even if you didn't see that, the parents each only has one phenotype. So if it was dihybrid, i.e. you're dealing with wrinkled versus smooth, you'd have two phenotypes for each parent. You'd have smooth and purple, let's say, and then you'd have wrinkled and yellow. Those would be your parents because they'd be homozygous and the opposite of each other. And so just from that information, you can figure out that it's kind of like, that's a shitty picture, hard to see, but you don't have to worry about it because the cross tells you that there are no wrinkled kernels in that image. So that was a long learning moment. Can't help myself. It's a professor in me, I guess. So for this one, you're just scoring color, right? So if it has the slightest bit of purple in it, like for example, can I zoom this in? This is such a shitty computer. Shouldn't really say that, but hang on, it's down here. Okay, so hopefully you can see it just a little bit better, even though it's slightly fuzzy now. So if it's got the slightest bit of purple, like these two here, right, they look a little yellowish, but they're not, they're purple, right? So even if you have even a little bit of purple, and this applies to the dihybrid cross too, then they're yellow, right? That's a good thing to remember. I'll just type that just for prosperity's sake. Right, remember there isn't an intermediate phenotype, so it has to be all yellow to be considered yellow. Again, you can see there's like some marbling up here and stuff like that. So all you need to do then is just do some counts and then come back to this cross. There we go. And work out what do you expect here? Actually, it tells you, but it wouldn't hurt to work it out. What do you expect in terms of number of purple kernels versus number of yellow kernels, right? Based on this cross. And so by doing that, that will give you your expected counts. So let's say you count 50, right? Then out of that 50 that you count, how many would you expect to be purple and how many would you expect to be yellow, right? You need to calculate that. Those are your expected values. And then you use your actual values, your observed data, and you carry out your chi-squared calculation, which is, and this is why these handouts are really handy, because they have all this information in here for you, right? So that's this. Slightly overcomplicated because I think it was written by a statistician. But essentially what it's telling you is chi-squared value over here on the left is the sum of the observed minus expected squared over the expected, right? And so down here, we're going to have two different classes. We have purple versus yellow. And so we're going to do two chi-square calculations. One for the observed purple, down here, minus the expected purple based on this cross, squared divided by the expected. And then add that to the observed yellow, right, these ones, minus the expected yellow squared over the expected yellow. Add those two together and that's your chi-square value. All right, so once you have that, oops, gone too far. Where am I at? Where did it go? hang on, where's... Oh, I'm sorry. Copying the questions from one to the other makes it harder to find things. You need to figure out degrees of freedom. So you remember that degrees of freedom is always n, which is the number of classes, minus 1, or number of different phenotypes, let's say, that you're looking at. And then you use either this chart up here, right, if that helps, and so again, you're going to be looking for the chi-squared value here, degrees of freedom, and then go across the intercept to see what your y-axis value is. Or use the table that we've used before. And so there's actually a copy of that in the exam and quizzes folder for today's exam. Oh, there you go. I guess you can guess that there's going to be a question on chi-squared in the exam, right? So, and then in Blackboard again, right, add in this, and then basically in a couple of sentences say, based on that chi-squared value and the degrees of freedom and then the p-value that you get, do you keep your null hypothesis, i.e. things are following Mendelian principles, right? And that's just a couple of sentences on that. So all of these questions are basically fill in the blank or short response questions. There's not really any multiple choice here. I think, yeah, I don't think there are. Maybe there's one or two in here. So there aren't really, there's not really feedback I can give you because it depends exactly on which kernels you score and how many kernels you score as to what exactly the chi-squared value that you get will be. I can't put in a value and grade you on how close you get to that because it's going to be different from one person to the other. And so just fill those out, basically. And then we'll do the same again for a dihybrid cross. Again, you got like some free points here, basically, because I'm telling the answer. E.g. question 1. Now you get the same pattern, right? Dihybrid cross again. Again, they do not change, right? So you always know that you're going to have homozygous parents, heterozygous F1s, and then a particular ratio in your F2. And so now, Angela, you can come back and look at this, and you can compare this with the picture above too, which is pretty neat. So we have the same parent, right? And so this is in our monohybrid cross up here, the parent looks the same, right? And so, but we're actually only concerned with one trait here, and that's color. When you look down at our dihybrid cross down here, if you looked, if I kind of put my hand just over the yellow one, that parent on the left, the purple one, looks exactly the same. It's only when you look at the other, which is the opposite, right? So if one is homozygous dominant, the other one has to be homozygous recessive. If you look at that one, now you can see that it's yellow, yes, but those kernels are not smooth. They're kind of like, eeh, I wouldn't eat that if someone gave it to me at a fair, right? No matter how much butter you put on it. They look wrinkled, right? They look kind of crappy, essentially. And so now you need to follow through the dihybrid cross again, just as you did in lecture. Look at the offspring, the F1s. You know what phenotype they should have and what genotype they should have based on your knowledge of how dihybrid crosses work, monohybrid crosses too. And essentially fill out those questions exactly the same as before. This time round, however, I've given you a better picture, and this should be a lot easier to use. Let's see, did I put that in? No, I didn't. I can upload that to the, let's see. Oh shit, I can't. I was a complete dumbass. Yeah, it's in the handout, but I was going to load it up to Blackboard as its own picture so you can blow it up. But then I remembered that I left my laptop in Georgia and it's not going to be here until tomorrow because I was a complete and total klutz. I'm doing this on my wife's laptop, which is why I keep insulting it all the time. Anyway, so that's actually a pretty decent high definition picture. And now let's see if we just blow it up a wee touch. Now you can see that, okay, we can actually see wrinkled here. And so all of these kind of crappy looking kernels, right, those are the wrinkled ones. And so now you can see you've got 4 different phenotype combinations, same as the key picture further up in the handout. You've got purple and smooth, you have purple and wrinkled, which are like these ones here. Again, if it's even slightly a little bit purplish, it's purple. It's a little trickier to see in the wrinkled ones because they tend to be darker anyway. But this one over here, for example, that I've got my cursor kind of circling around, that's yellow and wrinkled, right? Again, down here, you've got some yellow and wrinkled ones too. And you can figure out which ones you're likely to see the least of too, based on that dihybrid cross. Then you have yellow and smooth. So it's purple and smooth, purple and wrinkled, yellow and smooth, yellow and wrinkled. And you can see, if we go back up here, the parents only show 2 combinations of phenotypes. There are 4 in total, but you only see 2 in the parents. You see purple and smooth and yellow and wrinkled, right? And so that's the cool thing about dihybrid cross is that you see new combinations. Again, because those traits or genes are assorting independently, you'll see those. Get out of it. I don't know what I've done now. You'll see those traits mixing. So you're going to have 2 new combinations that you don't see in your parents. And those are going to be the yellow and smooth and the purple and wrinkled, right, which you didn't see originally. So all of those kind of give you some ideas about what numbers you should expect. And so essentially you want to do the same thing again. Count as many, how many did I tell you to count? Yeah, I think that was a copy and paste. At least 50 ears of corn? No. Don't do ears of corn. There's only 1 ear of corn there. Yeah, sorry. Kernels. Let's change that. That's better. Sorry, I think this was a copy and paste issue. I had lots of young screaming nephews and nieces running around while I was doing this. Yeah. There you go. That's what it should look like. So basically do the same again, at least 50, right? Because now you have 4 different combinations to work with. And so if you score too many, then too few, sorry, you might not get reliable data, particularly for the double homozygous recessive. And then you simply do the same thing again, right? You just do what you would expect, right? Again, this is based on the cross above here, right? What are the fractions of the total that you would expect to be purple and smooth, purple and wrinkled, yellow smooth, yellow and wrinkled, for example? And then you just use those to do the following questions. So how many how many chi-squared calculations are you going to be doing for this chi-squared? It's a question. 4. So how many degrees of freedom are you going to have? Yay! Exactly. Fabulous. Student interaction via miles of internet. Right, so just do the chi-square analysis the same. And now instead of, so either you'll use a different row in the table, or let's go back up here. Now you'll be looking to intersect with this line here. Hopefully you can all see that. That's the purple line with 3 on it, because that's your degrees of freedom. So either use the row for 3 degrees of freedom, or you use this line and it's the intersect for 3 degrees of freedom. So for example, if your chi-square value is 4, then you start down here, you go all the way up, and actually 4 is, oh no, because it's, again, 3 degrees of freedom, that's going to hit about there, give or take, and your p-value is going to be, between 0.2 and 0.3. So not significant, null hypothesis is kept, right? And your null hypothesis is that it's Mendelian, right? So if it's Mendelian, then you should get largely what you expect from a Mendelian dihybrid cross. So if you get a really high p-value, what would it be? Something around like 9 or 10 or thereabouts for the second one, almost certainly, and I'm not necessarily going to mark you down for this because it's the process that's important, but almost certainly, if I get down here quickly enough, you've counted some of these darker corns as yellow, right? And you shouldn't. So be very careful when you score these kernels that you're very sure there's no even little bit of purple there. Otherwise, particularly your yellow and wrinkled counts are going to be way off, right? And that's going to skew your chi-square result and make it look like it's significant. That's usually the biggest issue that in this particular lab that students have, right? If there's even the slightest, teeniest, tiniest hint of purple, let's see. Let's see what a good example would be. So that's a really manky looking bit of corn, to be honest. There we are. So this one, for example, here, right? I would count that as purple. Even though it looks a little bit yellowish, you compare it to the one down here, right? Or even the one up here, and it's darker than that. So just as with the smooth ones, there aren't actually many intermediate smooth purple ones here. Maybe that one up there, for example, right? That it's got a little bit of flash glare. But if there's even the slightest bit of purple in it, it's purple, right? And so if you do get a significant result, so a p-value of less than 0.05, it's almost certainly because of errors in counting. So you could always just go back and double check, or you could talk about that in your answer. And that's pretty much it. So it's really, oops, what did I do there? Who knows? I did something. I don't know what I did. But it's really fairly straightforward, right? It's very much a kind of practical exercise to reinforce the principles of Mendelian crosses that we did in class, and also give you some practice at doing a chi-squared. No, I don't want to do that. I think I just move this out of the way. I'll fix that later. Doesn't matter. It's the one that you've got. Who knows? Oh yeah, that's the one. I should keep you on retainer, Alyssa, as like a Microsoft technology expert. Oh, I didn't quite fix it. But anyway, doesn't matter. You get the idea, right? And that's it. It's a fairly straightforward one. It shouldn't hopefully take you very long to do either. Does anybody have any questions? No. And while I'm here, if you got any questions about the exam or anything else, I'm happy to answer them. Obviously, I'm not going to tell you the answers, right, to questions, but I'm here. All right, ladies and gents. Well, thanks for showing up and logging in. This is due for next Tuesday at midnight. Yeah, that's right. Tuesday's lab is already up, largely because I did that one first because I got them in the wrong order. I was like, ah, so I did both. And the audio recordings for the lectures for next week will go up, ideally, providing no other relatives have issues, by tomorrow afternoon, because I want to get that done and out of the way with. All right. So if no one's got any questions, thanks very much. Likewise, Alyssa and Angela, have a great weekend. Cool. Catch you later.